Hi, welcome to SDSF Live. Please let us know where you're logging on from. I'm Nicole Shen from Houston, Texas. We're in the middle of another tropical storm and hurricane in Houston. In the event I lose internet connection again, like last time, Michelle and Orva are still with you guys, but hopefully we'll be okay today. I was sad to miss the last show. My son Roman was in the hospital, so thankful Orva, who jumped on for me as co-host. The SDSF Live life is unpredictable. So glad we have a team of three of us to help each other. Our giveaway this evening is a fabulous SDS Strong mask made for you by our lovely Michelle. I'm pretty sure Michelle has made like 1 billion masks by now. Michelle, how are you today?
I'm doing good. We're doing really good up here. We are in like week 50,000 of the year 2020, and everything is going according to plan. We homeschool our kids, but we did add a couple of virtual classes in this year, so I kind of can feel everyone's pain. We're not used to having a schedule or wearing formal pants, and so this has been a big change for us. One of the rules for virtual was that the kids had to wear like, formal pants or shirts, like not pajamas. So that really threw us for a loop there. But we're surviving. We're doing really well. We're doing good. We're excited for fall. Michigan fall is, I think, my favorite season because it changes to really pretty colors. And you're right. I have made about 1 billion masks. I'm literally surrounded by masks, like every where I don't even, but here's some of the lovely ones that are ready to be shipped out to our amazing winner.
I wear mine all the time. I love it. Mm-hmm.
And they have, and depending on what size they need, I mean, I have all the sizes and I have backings and I have all the things.
It's not raining right now. It was raining a lot earlier and it was starting to flood. My yard was starting to flood, but we're fine right now. So hopefully I stay on and everything's okay.
Does that mean it stopped raining? I don't really know how hurricanes work.
So there's like bands that come through and it rains a lot and then it'll stop and then it'll rain a lot and it'll stop. So right now it stopped. Hopefully I'm okay.
I hope so as well. So the cool thing is today is that we're trying out a little bit something different. So we are still going with our SDSF Camp Live series because we wanted to bring as much camp to you guys as we could. However, we're also broadcasting it to YouTube now at the same time as Facebook. So I wanted to show
over here.
We're getting real fancy. Still haven't gotten that podcast, but getting real fancy everywhere else. So I wanted to show you guys the easiest way to know when we go live. First, I will always make an event so you guys can subscribe to the event or hit interested or going. And that's how you'll get updates on it. We've had to, push a few, there was some time changes. To know when we're going live, you can either go to YouTube and subscribe to the SDS Foundation page. And then that little bell next to subscribed will alert you when we go live or post a new video, as well as on Facebook. If you click subscribe onto the Facebook or like the page under settings, right there on that picture, there was a you can click to be notified first. So it'll show up in your news feed. Thank you, Orva. So it'll show you first when we're live. So hopefully that helps. We had a couple of emails come in and people didn't want to miss the shows coming up. And so we do have a couple of really good ones coming up after this one. And I'm kind of excited. So you can win the mask that I was holding up by interacting with our show in any way that you've done before. You can like, comment, share, start a watch party. Those are really, really, really cool to do. You could share the YouTube page, probably. I don't really know the new rules for the YouTube one, but I don't know. Yeah, no. Like Nicole said, when you guys log on, let us know where you're watching from. Recently, Orva got an email from someone out of the country, and I can't remember where. And I'd gotten an email from someone in Germany, and then one from Italy. And so it was really cool to see that.
Really cool.
This is helping people who camp wouldn't normally reach. So this evening, we have a return guest coming on, Dr. Seth Corey from the Cleveland Clinic. Right tonight, he's here to present his latest research with us. And if you're not familiar with Dr. Corey's research, he has been researching the studies of effects of SDS in zebrafish, which has been really, really, really interesting to see what it looks like. And I won't ruin any more because I don't know the most of it like he does. So welcome back, Dr. Corey.
Hi, Dr. Corey.
Greetings from Cleveland, where the weather is getting chilly, but we have no hurricanes coming towards us.
So we were just chatting before the show that Dr. Corey used to work in Houston at MD Anderson. So he's very familiar with the Houston weather and craziness here.
Maybe I should move back, because when I was in Houston, I never had a hurricane.
Maybe it's the Corey effect.
Maybe, and I was in New Orleans for medical school, and that's also in the hurricane, as people know terribly, tragically from Katrina. But I actually grew up in Orlando, Florida, where we did have hurricanes.
Yeah.
And we had to board up the house and do the tape and, get water. So,
well, Dr. Corey, we would welcome you with open arms if you wanted to come back to Houston. So maybe you should come back.
I like Houston. We have no one from Cleveland, but Houston's great, especially when the rodeo comes back and South by Southwest. And hopefully coronavirus will not last beyond 2020. Okay, but we're not here to talk about coronavirus.
No, much like the last couple of shows, we're actually going to have Dr. Corey share his screen with you guys, so you're not even going to get to see Nicole and I that much, which is totally fine. He's going to share the presentation and then we'll take questions afterwards. So please type your comments or your questions in the comments, and we can read them as they come in, and we will make sure Dr. Corey gets to see them. So the floor is yours, Dr. Corey.
Okay, so actually, I'll talk a little bit about our paper that appeared in Journal of Clinical Investigation Insights last month. It's available, I believe, freely. I have sent a copy, I think, on to Joan. And if anyone emails me at corylab@yahoo.com, I'll be happy to send them a copy of the publication. It's about 15 megabytes. So if your email can handle 15 megabytes, I can send that out. This is really the work of a really great scientist in our lab, Dr. Usua Oyabide, who's now an assistant professor. And she's worked about 6 years on this project. So it's a long pregnancy that finally gave birth. And we're working on the follow-up papers. So I'm going to switch over and let me know if this is working. Okay, share. All right. And let me see. Oh, it's not that bad. I hope somebody wasn't screaming in the background. Can people see the PowerPoint?
Yes, we can see it, Dr. Corey.
What happened there? Okay, so this is this next Tuesday. I'm giving what we call grand rounds at the Cleveland Clinic Department of Pediatrics, and I will be talking about our work on pediatric leukemia as a complication of the inherited neutropenias. I'm interested in the inherited neutropenias, and with special focus, almost exclusive focus, on severe congenital neutropenia and Shwachman-Diamond Syndrome. And so it's entitled A Gain of Clones, Pediatric Leukemia Predisposition Syndrome. So the objectives to the group that I'm going to talk to on Tuesday, and I guess my objectives in talking to you tonight is to talk about not really the inherited bone marrow failure syndromes, but to focus on Shwachman-Diamond, to try to convey what are risk factors for the transformation of marrow failure to leukemia and cancer. We know this far better for severe congenital neutropenia than for Shwachman-Diamond syndrome. And that really has been one of the major interests, is to try to understand who with Shwachman-Diamond syndrome will be at risk for developing leukemia or its related condition, myelodysplastic syndrome, with the idea of trying to prevent that through an intervention. So some of the questions we're interested in, and feel free to interrupt, okay? So the questions we're interested in: what causes leukemia or pre-leukemia, which is a term to describe myelodysplastic syndrome, which overlaps considerably with acute myeloid leukemia, but in and of itself is not leukemia. Who will get leukemia? Can we identify risk factors early on, before, if you will, the horse leaves the barn, before the chemo-resistant clone becomes established, which at that time makes treatment by chemotherapy and or stem cell transplantation all the more difficult. But not every child, certainly not every child or young person with Shwachman-Diamond Syndrome needs a bone marrow transplant, a stem cell transplant. And the idea is to understand what are the cooperating mutations, what are the processes that sort of trigger the transformation from an inherited bone marrow failure syndrome like Shwachman-Diamond syndrome to a myeloid neoplasia, either acute myeloid leukemia or myelodysplastic syndrome, and how can we intervene more effectively, with the goal really trying to avoid stem cell transplant and to avoid transformation. So this is sort of more broadly for the pediatric audience. You know, one of the things that we're beginning to learn is that family history is very important. This is probably not the case for Shwachman-Diamond syndrome, but in families that we see, if there's 2 or more blood cancers or cytopenias, which is a shortage of one of the cell types or several of the cell types, that is white cells, red cells, and platelets, in 1 or 2 relatives that really go back to the individuals, like grandparents, over 3 generations, then that's a clue that there may be some sort of genetic predisposition. And what we're learning is that in about 10 to 12% of adults, and in about 10% or so of children, that there is what we call a germline mutation, a variant in the code for a particular gene, for a particular protein, that differs significantly from the rest of the population, and that it carries with it a pathologic significance. And what's interesting, certainly to some of my adult colleagues, is the fact that some of these genes, when mutated, don't manifest as myelodysplastic syndrome or acute myeloid leukemia until much later in life, even into the 7th decade of life. And some of those genes are listed here. But what is interesting and is relevant to the Shwachman-Diamond Syndrome Foundation is that there is more than a handful of patients that have been diagnosed with myelodysplastic syndrome or acute myeloid leukemia in their 30s and 40s, who, when investigated, were in fact individuals with Shwachman-Diamond syndrome. They just were not diagnosed at an earlier age. So the issue of hereditary hematologic malignancies is a growing one in appreciation and in diagnosis, especially being afforded now with next-gen sequencing. So cancer is a genomic disease, meaning that cancer results from multiple genetic variants. These variants can be a mutation, a change in the code. It can be a shuffling of genes between different chromosomes or within the same chromosome. There can be amplification or deletion. We call that copy number variance. Or there can be, for instance, a virus that inserts into a gene or in its regulatory component. And so these genes that cause cancer can be classified either as a proto-oncogene or a tumor suppressor gene. And this presentation is geared towards pediatricians. And one of the messages is that we can learn a lot about adult cancers by focusing on pediatrics. And in fact, this picture here is a picture of Herb Abelson, who was chair of pediatrics at University of Chicago and University of Washington, who as a fellow back in Boston discovered a retrovirus gene and the gene was named Abel after Herb Abelson. And that's the gene that's involved in chronic myeloid leukemia, the so-called BCR-ABL. So there are oncogenes that result from these mutations, which is like a gain of function or putting a foot on the accelerator and causing the cell to divide over and over again. Then there are genes that turn off the growth of cells, sort of like putting a brake on the car. And these tumor suppressor genes, when mutated and associated with cancer, result in their loss of function. So you don't have that brake acting on cell growth. And this entire field of tumor suppressor genes came from a relatively rare pediatric cancer called a retinoblastoma, a tumor in the back of the eye. And you can see that this young girl has a tumor that you can see in the back of her eye. And another pediatric hematology oncology fellow who was a couple years ahead of me back in Boston, Steve Friend, cloned the gene that's responsible for this, and it's called retinoblastoma. And it's a gene that's involved in this very complicated picture of genes and proteins that control cell division. So cancer is a genomic disease resulting in changes either in normal genes called proto-oncogenes or tumor suppressor genes, resulting in either a foot on the accelerator or the inability to cause a brake. And cancer as a predisposition disease is becoming better and better recognized, that these variants can be somatic, but these variants can also be germline, that is inherited either from a parent or perhaps more commonly as sort of a new mutation that occurs very early on following conception. And so there are a variety of genes that are recognized as being germline, and these are of importance both for kids as well as for adults. And one that you may be sensitive to are the genes that are involved in breast and ovarian cancer. One of the more common ones being something called breast cancer antigen 1 or breast cancer antigen 2. Some of these mutations, such as the genes causing neurofibromatosis and ataxia-telangiectasia, can actually result in a syndrome, just like Shwachman-Diamond Syndrome, which represents a mutation that affects multiple tissues. And we're able now, through very powerful tools called next-generation sequencing, to be able to screen tumor cells and to screen non-tumor cells, looking for genes that are mutated, looking at a whole battery of gene panels for instance, bone marrow failure. Although relevant to Shwachman-Diamond syndrome, some panels don't contain the gene SBDS, which is the gene that's responsible for about 90%. I know one company, Invitae, has had trouble trying to get a accurate test up and running, in part due to some difficulty in developing the tools to detect mutations in the Shwachman-Diamond gene. And one of the problems that we're beginning to face is that as we do more and more next generation sequencing, that we now are identifying either single nucleotide variants, which may or may not have any pathogenic significance, or variants of unknown significance, genes that have a difference in their code in a small number of individuals, but it's not clear whether those mutations or those variants have any pathogenic disease-related significance. So we call that variants of unknown significance. Now, in the case of Shwachman-Diamond Syndrome, this is an autosomal recessive condition where you get one gene from one egg and another gene from the sperm, and that you need to have both genes vary from the normal gene in order to result in the disorder. But another way to look at all this genetic information is that disease is really about probability. Just because one has the mutations or the variants, either in a single dose or a double dose, does not necessarily mean that everybody has the same appearance. So there's the genotype, which is the genetic information, and then there's the phenotype, which is the appearance. So not all kids with Shwachman-Diamond syndrome look the same, act the same as far as their complications or their situations. And we don't fully understand, what are the factors that cause the phenotype to vary between individuals. But we may consider this as sort of deterministic and stochastic factors. Deterministic factors is actually the instruction of the Shwachman-Bony and Diamond syndrome gene having one of the mutations. And the stochastic factors are sort of probability events. And some of the things that affect what we call the penetrance of the disease, the probability that a disease will manifest, would be things such as what we call epigenetics, which is modification of the genes that's to be expressed. That would be modification of the Shwachman-Diamond gene expression. It could be the environment, which we're very much interested in, and that is there exposure to a virus or to a bacteria? Is there exposure to a chemical that or a dietary factor that can modify the disease? So there are exposures in the environment. And then something called epistasis is really a fancy term to refer to the fact that there may be other genes that are co-modifiers. So there's about 19,000 genes in the human. And Shwachman-Diamond gene is just one of those 19,000. And it could be that one of the other 19,000 or so genes might modify the course of the disease. And in cancer, there's another factor that takes place, and that is the emergence of clones of cells. And whether these clones of cells that arise, like clones of cells in your bone marrow, whether they emerge, whether they become extinct, or whether they become dominant, and whether they become dominant enough to cause a leukemia. Everybody's quiet. Are you following me?
We're here.
Okay. I haven't put anybody to sleep yet. I can't see anybody, so I don't know if I put people to sleep.
No, nobody's asleep yet. Joan did ask, though, and I think she's referring to the genes you were talking about in the beginning. Are these genes seen in SDS patients?
Okay, so I think the answer is those are being, like RUNX1, GATA2, and P53.
Yes, I believe that's what she was talking about.
Yeah, I apologize. When I switch over to the PowerPoint, I don't see the chat box, okay?
No worries.
So the answer is yes, and as it's somewhat controversial about what the role of P53 is. So P53 is the guardian of the genome. It's a repair gene. It sort of tells the cell if the cell becomes stressed for one of any number of reasons, one of which might be ribosomal stress, to sort of pause and to repair itself before undergoing cell division. And so there is an association of P53 mutations with Shwachman-Diamond syndrome. And the data are somewhat controversial. There's the group from Boston with Lindsay Coleman, who's found a very high correlation between P53 mutations and the development of myelodysplastic syndrome or acute myeloid leukemia. On the other hand, the Severe Congenital Neutropenia Registry reported that there were P53 mutations that were isolated in individuals with Shwachman-Diamond syndrome, but those individuals did not have any blood cancer. So one can have a mutation in P53. It may or may not be a harbinger of transformation. That's what we're trying to figure out in our zebrafish. And that was sort of one of the first reasons why I started off on this project in working on zebrafish, was to look at cooperating mutations. We've sequenced about 7 patients that we obtained from doctors in Italy, Marco Cipolla, and we didn't find any pathogenic mutations out of 7 individuals that had acute myeloid leukemia and Shwachman-Diamond syndrome. I'm not aware of RUNX1 or GATA2 mutations. There is a paper that has been deposited in the bioarchives from Lindsay Coleman to suggest mutations in EIF6, which we'll get to later on. So as we do more and more candidate gene sequencing, there will probably be variants identified in these genes, as well as other genes associated with hematologic malignancies. Whether they play a causal role or whether they are causally associated, that needs to be worked out. And so I don't know if that answers Joan's question, but I think the question that we would have is that if one does detect a mutation in P53 in the individual, what does one do? And I'll get to that with, I think, my last slide. Okay. Any other questions?
I don't believe so yet. I know I'm taking notes.
Okay. I'm going to skip over the severe congenital neutropenia part and get to Shwachman-Diamond because I don't want to take up too much of your time, and everybody's really interested in Shwachman-Diamond syndrome. But before I sort of segue into that, this is just a list of the more common inherited bone marrow failure syndrome. Shwachman-Diamond syndrome, is definitely one of the more common ones. The epidemiological data suggests that it may be as common in 1 in 75,000 births. If one added up all the Fanconi anemia, severe congenital neutropenia, Diamond-Blackfan anemia, dyskeratosis congenita, there's about 1 in 30,000 births with one of these leading inherited bone marrow failure syndromes. And so FA is Fanconi anemia, severe congenital neutropenia, Shwachman-Diamond syndrome, Diamond-Blackfan anemia, congenital amegakaryocytic thrombocytopenia, dyskeratosis, congenita. And some of these have multiple lineages in the blood system affected, that is white cells, red cells, and platelets. Others are more lineage specific, such as Diamond-Blackfan anemia that affects red cells, or congenital amegakaryocytic thrombocytopenia, which affects platelets. In the case of Shwachman-Diamond syndrome, is from your own child or friends, relatives situation, that predominantly neutrophils are affected, and that can fluctuate. and then it can be either severe neutropenia or more mild, moderate neutropenia. And in a handful of other patients, individuals, you see anemias and thrombocytopenias. And I had one child who had transfusion-dependent anemia that resolved by age 1. I don't know whether, and never reappeared again. The incidence of myelodysplastic syndrome or acute myeloid leukemia, and I'm not here to frighten you, but data from Blanche Alter, and in particular the Severe Congenital Neutropenia Registry in France with Jean Donadieu, suggests that the incidence of Shwachman-Diamond syndrome transforming to myelodysplastic syndrome may be as high as 30 to 40%. And we don't really know the full or the most accurate lifetime risk, because there are individuals that are not diagnosed. There are individuals that are diagnosed as a result of being evaluated for myeloid neoplasia. So that might be what we call a wash. There's something called reporting bias, that those individuals with Shwachman-Diamond syndrome that have myeloid neoplasia, AML or MDS, are more likely to be reported. So this is, one of the goals of the Shwachman-Diamond registry, is to try to get a more accurate handle as to the percentage of individuals that are at risk for MDS and AML. If you had to ask me, I would probably say that the risk is less than 30 to 40%. There are anecdotal reports of breast cancer and pancreatic cancer, but this appears mostly rare and, as I said, anecdotal. And what's interesting is that these inherited bone marrow failure syndromes involve particular pathways that are fundamental to cellular behavior. In the case of Shwachman-Diamond, it's really the ribosome, which is a machine inside the cell that's responsible for making protein. Okay, so let me skip over this part and get to...
Dr. Corey, while you're skipping, Joan had asked, where did you get the stats of 1 out of every 175,000?
1 in 75,000. 1 in 75,000 is from the Italians.
Got it. And then she had a follow-up question of, and I don't know quite what she was asking it to, but do they always progress from MDS to AML?
That's a good question. The answer is not necessarily, because by the time they develop MDS, which is the definition of myelodysplastic syndrome, is peripheral cytopenia, and many of them already have that, with dysplastic features, which is a term for structural abnormalities that's found in about 10% of a particular cell type. So that could be a weird-looking nucleus, which is the inside of a cell that contains the DNA, or it could be the granules that contain some of the protective enzymes don't look normal, and it's oftentimes the dysplasias in the eye of the hematopathologist. In adults with myelodysplastic syndrome, about a third of them will go on and develop acute myeloid leukemia. If I had a kid with myelodysplastic syndrome with Shwachman-Diamond syndrome, the standard procedure would not be not to wait till it transformed into acute myeloid leukemia, the definition being around 20% blast. So it's a sort of an operational definition. The difference between myelodysplastic syndrome and acute myeloid leukemia is 20% blast or more in the bone marrow. So if you had a child, for instance, with myelodysplastic syndrome or Shwachman-Diamond syndrome that had 5 to 10% blast in the marrow, we would call that myelodysplastic syndrome secondary to Shwachman-Diamond syndrome. And the treatment of choice would be to go and have a stem cell transplant. So, I think the natural history of how many individuals that will transform with MDS to AML is not clear, and I think current practice would sort of prevent a very accurate accounting of that. The other thing, as you all know, is that the presence of a cytogenetic abnormality in an individual with Shwachman-Diamond Syndrome may or may not be associated with transformation to a life-threatening myeloid malignancy. So you can have individuals with deletion 20q or isochromosome 7 who do not, who appear to have stable disease. And I think that's something to be debated among the transplanters, is whether that's an indication to transplant or just to watch conservatively. On the other hand, if an individual has evidence of loss of chromosome 7, that would be a marker either for myelodysplastic syndrome or acute myeloid leukemia, and that would clearly be an indication for transplant. So I guess that's a roundabout answer to Joan's question. Does MDS evolve to AML in kids with Shwachman-Diamond syndrome? It can. As far as the absolute risk, it's hard to ascertain. That the current approach would be to intervene with a stem cell transplant before you hit that 20%, which is a marker for an operational definition of acute myeloid leukemia. Any responses?
No responses to that necessarily, but Joan did say that she recently read that the Italian latest stat was that is 1 in 150,000 births for SDS.
Yeah, I haven't heard that from Marco or Valentino. Those numbers are, according to a mathematician modeler, you can be off by a log, which is a factor of 10, and it doesn't make the models that much different. Whether it's 1 in 75,000 or 1 in 150,000, it is a rare disease. But if you add up all the inherited bone marrow failure syndromes, these are epidemiological studies from Denmark as well as from British Columbia. They will approximate the number of patients with newly diagnosed pediatric AML. It's a rare disease.
Joan had a really good question. Joan's full of them, and I love it. She said, have you seen any of the zebrafish develop MDS, or is that even possible?
I'm not going. I was looking for the slides to add to the presentation. To get to her question, no, we have not. However, issue number 1 is our fish die somewhere around 25 days post-fertilization, which is early childhood. However, we have put in the R126T mutation into the fish, and so that's in the background of not having any Shwachman-Diamond gene that's functional. We've made fish that express the R126T, which has been associated with human disease, and the fish they've lived as long as a year. And we're analyzing the fish now. And interestingly, they appear to sort of catch up. Initially, there's some decrease in length of the fish, but within a couple months, they reach normal size, or a size that's no different from their, if you will, normal siblings, that there's no gross evidence of any cancer or blood disorder, but we're just beginning to analyze these fish as far as their neutrophil counts. There is, in some cases, some curvature of the spine. That's a rare event. The fish are fertile, so both boys and girls can lay eggs. And so we're analyzing those fish now. And we've made human rescue as well. And so Sue is making a bunch of these different sort of partial rescue fish. So to answer your question, in the knockout fish, they die by about 25 days post-fertilization. We've been able to rescue the fish using an abnormal protein, but it's abnormal. It's not so abnormal so as not to cause disease in the fish. And so and we have some evidence that they do have mild activation of P53 as well as some other genes. So the conclusion from that is that if the zebrafish are an accurate model for Shwachman-Diamond Syndrome, and I think they are, then there needs to be cooperating genes that have to be mutated to cause leukemia. And so we're trying to interrogate those by looking at things like EIF6 and P53. Yeah. And we're also stressing the fish with ultraviolet and other chemicals so as to try to induce more genetic instability that will cause variants to arise in critical genes. Okay? Are you guys awake? Hello? I lost you? Houston, we have a problem. Are people there?
Can you hear us, Dr. Corey?
Yeah, I didn't know whether.
No, you're here.
Okay. You know, I can talk about this for hours, and you know, I know you all are very interested in it, but let me try to go over this a little bit more quickly. But feel free to just tell me to shut up and move on. So we'll skip this slide, other than to highlight the fact that there are other genes that have been identified with overlap syndromes, the elongation factor like 1, and a gene called DNA JC21, and a gene called SRP54, which has overlap with both Shwachman-Diamond syndrome as well as severe congenital neutropenia. Okay, so, okay, all right. So, the question is, you know, how does one study a rare disease? And to identify phenotype, for instance, the emergence of leukemia that may not be present in a very high percentage. So 30, 40% maybe, but maybe it's less than that. So you can use mice, but mice have small litters, several mice per generation. They're very expensive to maintain, and the problem is that when the gene was knocked out by Joanna Romans in the mice died very early, sort of in the first trimester. And so that means you have to make something called a conditional knockout, which makes it even more expensive and more cumbersome. But when we first started this project, and I don't even want to tell you how long ago I first had the idea of doing this, but it was basically around 2004, 2005. And we sort of had to wait for the technology to catch up. The zebrafish protein, so genes make protein, and there's about 22 different types of building blocks called amino acids, and they all have single letter abbreviations. So if you can just take a look at the red, and all the red indicates how well conserved the protein is between human to rat to chicken to snake to frog to fish. And so you can see that the acid is pretty comparable, pretty highly conserved between human, and so that told us that we could try to knock it out. Okay. My computer's acting goofy. Okay, so one of the things I want to emphasize that the zebrafish is good for, besides the fact that it's conserved, is that a fish will lay hundreds of eggs. So that means we can look at large numbers of offspring and be able to do statistical analysis, and that we can genetically manipulate the fish. And there's a new technique called CRISPR DNA, and letting the DNA repair itself, but it's not 100% successful. And in that 1% or so, you will develop what we call deletions or insertions. And the SUA did this about 5 years ago, and she was successful in making 7 different strains of fish that lacked the Shwachman-Diamond gene product. And we could see that the little green specks are neutrophils, and we can see that the fish that don't have the Shwachman-Diamond gene have far fewer specks. When we looked at macrophages, which is a normal number between the affected fish and the normal fish. When I say normal, I'm talking about wild type, the ones that have the diamond. And we looked at red cells by staining, and this is not so quantitative, but more qualitative. And you can see 2 things here, okay? One is you can see the size of the fish, and you can see the organs. There's the heart there. The sizes between the 2 fish at 5 and 10 days post-fertilization and red are the same between the fish. We did see that there was some bony defects, and this was noted by Asua when she would cut the fin to get the DNA, all right, then the fin wouldn't regrow. And the fin regrows by migration of these bone-causing forming cells called osteoblasts. And when we looked at the fish that were stained with a dye for bone, if you have really good eyesight, you can see defects in the spine and some curvature of the fish. So we've seen some scoliosis, curvature of the spine, in some, not a high percentage, of fish that have survived beyond 25 days of fertilization. We looked at the kidney, which is the source for blood cell production. It's the bone marrow equivalent. And we saw no differences. Between the wild type and the mutant. We looked at the pancreas, and we can see at day 15 that the pancreas in the top part is one part affected. 21. Okay, you can see here. See the shrivel up. So their pancreatic also began to be smaller in length. So they were smaller, nutrient, and pancreas having atrophy by 21 days post-fertilization. And what's really interesting is their intestinal tract, okay, shows blunting of the intestinal sort of lumps and bumps that facilitate absorption. And you can see the amount of space in the intestine in the normal fish is much contracted in the affected fish, okay? This is the food particles that they eat. So you have atrophy not only of the pancreas, but you have atrophy of the intestinal tract. And there was a report, I believe, from a Scandinavian group that did endoscopies on kids with Shwachman-Diamond Syndrome. And about half of the kids, they did find evidence of intestinal atrophy. I would be kind of curious as to whether any of your children have had issues with such that they had endoscopy done, a tube, snake through either their stomach or their butt to take a look at the intestine, and whether there was any evidence of intestinal atrophy in your children.
Dr. Corey, Joan had asked, are there any organs in the fish that
So this is one of the
not
advantages
affected by
of having a
SDS?
fish model, a whole animal model, is that we can take a look at, for instance, the eye, and we can take a look at the intestine, and we can take a look at the heart, and we can take a look at the brain. We have looked at the brain, for instance, and grossly we don't see any changes in brain structure between the wild type fish and the fish that have mutation in the gene. Okay.
Dr. Corey, can you hear me?
So, Ed, what we've done is to take the fish and do something called RNA-seq, which is to take all the messages for genes and to sequence them and to look for genes that are upregulated or downregulated.
Dr. Corey?
In
Hey everyone, thanks for hanging in with us. We are working out some technological issues that we are having on a couple ends here. We're trying to get Dr. Corey back. We were trying to ask him some questions, and obviously there's some form of, I created a verb for this, Nicole. I don't know if you use it. I call it 2020.
Talking to Coach, she has interest in TG pathway, but we didn't see much in the way of TG. Instead, we do what we call the amount of gene message post-fertilization to 21 days post-fertilization. You have upregulation of genes that are either involved in DNA repair or in cell death, like this caspase and Bax and Puma. And what's really striking is that P21, which is a gene that's a tumor suppressor, is tremendously upregulated 80-fold by 21 days post-fertilization. And what's interesting, in the fish that we've observed that have survived for a year, we do see increase in P53, not so great, only a couple-fold increase. But again, the P21 response,
For some reason,
go
we
ahead.
can still hear him. He's no longer on here.
Oh, Dr. Corey, can you
Are
hear
you
us?
there?
Dr. Corey?
Hello? Yes?
So your audio keeps cutting in and out a lot. And then at one point it was like your slides disappeared and then you disappeared. And then, so we were trying to get you back to where you could hear us, because I was asking you a question and it was just like, I don't know if you could hear us at all.
The audio keeps
There you are.
cutting
We
out.
seem to be having some technical difficulties.
Can you?
So I think what we should do, Nicole, is maybe let's throw Dr. Corey's presentation up again, and then
Can you hear
it
me?
seems to be that we can hear him when he's on his presentation, but he's having a hard time hearing us. So we'll let him finish his presentation and then try to ask questions at the end.
So he's actually no longer sitting in his chair.
Dr. Corey has left the building. Dr. Corey said, I am done with this technical difficulty of 2020. I'm out. I have left my chair.
I'm not too sure what to do right now.
Well, I think, so is he still in the broadcast at all, Orba?
Not anymore.
Not anymore? Okay, I'm wondering if maybe, because it sounded like it was getting spotty and then like his internet was cutting in and out. So maybe we'll give him a couple minutes to come back and see if he wants to finish up his presentation. And yeah, he timed it almost perfectly for internet issues. We're at about an hour, so.
I'll try to email him real quick.
What was that, Nicole?
I'll try to email him.
I thought it was interesting. I knew about his zebrafish research, but hearing the ins and outs of exactly why they choose the zebrafish always fascinates me.
Hi,
Dr. Corey, welcome back.
I was falling asleep. Anyway, so I'm sorry, but all of a sudden everything went dead, and I see Joan asked, "Are there any organs of the fish that are not affected by Shwachman-Diamond?" Well, the kidney, interestingly, is not.
Oh.
At the microscopic level, and that's the area that that blood cells are produced.
Mm-hmm.
As I said, we haven't had a neuropathologist take a look at the brain, but the brain appears grossly normal. The retina, which is the back of the eye, I'm curious as to is profoundly affected. Now, it may be that the eye is a major organ in the fish, but I'm curious as to whether any of your kids have had visual problems. I noticed some of us wear glasses, but I think that from insight, and it may not pay off, would be to have an ophthalmologist take a look at the backs of eyes in a regular, intensive way. I don't know whether you heard me, but there was atrophy, which is sort of a failure in growth of the cells that line the intestinal tract. And there was this paper from Europe that suggested about 50% of kids with Shwachman-Diamond syndrome had intestinal atrophy, which may compound their malabsorption and contribute to the nutritional stress, nutritional deficiency.
Dr. Corey, is that the same thing as gastroparalysis?
Gastroparalysis or gastroparesis means that the stomach does not pump as well.
Okay.
We have looked, now there's skeletal muscle and there's smooth muscle. We have looked at skeletal muscle, so that's like the muscle in your biceps, okay? And the muscles appear to be the same. But the muscles that cause the contractions of the stomach or the intestine, they're smooth muscle. So, not to go into patient confidentiality information, but I will go back, take a look, and see if there's something that's an abnormality in the smooth muscle in the fish. Okay.
My son has gastroparesis as well as some intestinal issues that I will happily send over to you so you can take a look at them, because he's always kind of, and I know quite a few SDS patients have, delayed motility type issues, weird things that will show up on a biopsy, intestinal wise. So it's interesting to hear you say that there's some of that kind of showing up. We always suspected that Nathan's was sort of comorbid with another syndrome that he has, but we've never really been able to sort the needle in the.
Well, if people want to communicate with me sort of confidentially, but does medicines like metoclopramide or Reglan work at all?
Not for Nathan, no. I know for some SDS patients that it does work. With Nathan, it produced the side effects weren't worth it for us. So it may have worked as a medication, but it didn't work overall.
So and I'm not an intestinal biologist, but the intestine has these little crypts of cells that project into the space of the intestine. The fish have one intestine. We have two types of intestine: small intestine and big, and the colon. Okay, large intestine. And there's different compartments in mammals. You have the duodenum, jejunum. Okay, and so there's specialized areas of nutritional absorption. For the fish, there's just one type, and I'm not aware of further compartmentalization. So, what we do see is epithelial, or the basic intestinal atrophy. And the motility can be a result of smooth muscle, which is outside of the intestine, okay? And we can take a look at the amount of smooth muscle. There's also sort of innervation, the nerves that go to the intestine, all right? And so we can take a look to see whether there's any abnormalities in the smooth muscle or in the innervation of the intestine. But the eye is definitely the retina, the back of your eye, that has all the rods and cones that pick up light and transmit it through the optic nerve to your brain. That's clearly has atrophy. I didn't include that picture. And a lot of people use zebrafish to study eye development. So I would be curious as to, whether anyone has looked at an ophthalmology exam, in kids with Shwachman-Diamond to see if there's been issues with that.
So what does that translate to ask an ophthalmologist?
I'm sorry, what about an
What does that translate to ask an ophthalmologist, like on an eye exam?
To see, the backs of the eyes, to look for any, thinning of the retina, which, they would have to do that either examination under anesthesia or a slit lamp for somebody who's cooperating, to do dilate the pupils and to look at the back of the eye through a slit lamp exam. And to do this sort of in a methodical way, to look at a number of kids, for instance, to get an idea.
Is there an age you would recommend that for?
Is there any what?
Is there an age you would recommend that for?
I'd hate to expose a child to anesthesia. Okay, so I guess I would, unless there's some basic visual checks that identify issues, I would probably wait till preschool, first grade, second grade. Yeah.
Or maybe it can be combined with another procedure?
It could be, yeah. I mean, it could be like a bone marrow exam or endoscopy. I guess the issue is, and I get to that to the last slide, is sort of what really constitutes optimal surveillance of the children. There's surveillance to gather information, and then there's surveillance to develop interventional strategies, okay? And different parents may have different views as to how much they want to subject their child to sort of surveillance to gather information. And I think most of that, that would have to be a protocol. That should be a protocol with appropriate informed consent.
Yeah. That's really interesting. That's new to me. That's really interesting development.
Yeah, and in turn, if if individuals, parents. So the idea in research is to go from the bedside to the bench and back to the bedside. And so any sort of clinical insights that we get from the children, from you all, for instance, like some have noted about ADD, hyperactivity, then, what can we go back to like an animal model and to understand what's going on? Clearly the neutropenia, clearly the pancreatic atrophy, clearly the intestinal atrophy, the bony abnormalities, and there's quite a variation in bony involvement with kids with Shwachman-Diamond syndrome, from the trivial to the life-threatening thoracic dysplasia. Then we can go back to the fish or the mouse, if that is the situation, and ask questions. So, I'm sorry if I'm a little bit verbose, but is any other questions? And I can try to sort of finish up quickly, because the kids are wanting your attention. So what we do have now.
Do you want to try to put up your last couple slides, Dr. Corey?
I hear you.
Okay,
yeah.
So let's put up.
I'm going to try to do that. Okay, and see how I can, because I know it's late. So here, okay. So.
I mean, I have all the time in the world.
This is getting late, and you've got kids to put to bed, and let me show you sort of the grand unifying hypothesis, okay? Let's see here. I'm trying to share my screen. Okay. And here we go. So, do people see this picture?
Yes.
Okay.
Yes, you are loud and clear.
Okay, but you see this diagram. People see the diagram?
Yep.
Okay.
Yep, we can see it.
Okay. So we're trying to put together a sort of a timeline for development and how development goes off the pathway for kids with Shwachman-Diamond syndrome using a powerful animal model, the zebrafish. And so in going left to right, at the time of fertilization, you can thank your mother, meaning the kids should thank you for giving them sufficient amount of both the message to make Shwachman-Diamond gene, as well as having protein available from the mom. But the fish survive, we think, in part because their development is quick and they get a sufficient amount of protein and message from the mom, and that levels don't disappear to around 8 days post-fertilization. And as the declining amount of Shwachman-Diamond protein occurs, then we start to see stress responses among the different components of the ribosome, and then responses by the p53 pathway that cause cell cycle arrest, and metabolic abnormalities as demonstrated by some of these enzymes here, decreased amount of fat that gets absorbed, an increasing amount of genes that express proteins that are involved in apoptosis or a cell cycle arrest. And then at this point, after these early biochemical and genetic changes have been detected through the power of the zebrafish model, then we can take a look between 15 to 21 days post-fertilization, early childhood, if you will, and we can see atrophy of the eye, of the pancreas, of the intestine, and organ failure. And then regrettably, the fish die around 25 days post-fertilization. And my hypothesis is that in addition to these abnormalities, what we're also seeing is a vicious cycle where there's decreased nutrition and increased tissue injury, and that leads to the fish dying. And we're able to rescue this now with both putting in the human gene as well as the mutation. So, the issue, that we were just talking about is, not just surveillance for children with leukemia, but, how does one survey, intestine and heart and growth? And for, unless I hear otherwise from you all, 'cause I'm not a parent of a child with Shwachman-Diamond, and each one has different, concerns. I mean, to me, I guess my concern would be, I would be worried most about the transformation to myelodysplastic syndrome or to acute myeloid leukemia, because those are truly life-threatening situations and results in premature death. I also appreciate in hearing from the Shwachman-Diamond Foundation that individuals that have survived in the 5th and 6th decade of life are beginning to unfortunately die from non-alcoholic steatorrheic hepatitis or fatty liver disease. And one of our interests is to take a look at the liver and see what's going on metabolically that results in sort of these early, late deaths from individuals affected with Shwachman-Diamond syndrome. And all this tells us about the biology and the natural history of the disease. But eventually what we like to do is to try to develop some sort of therapy. You can do stem cell transplants, but not everybody needs a stem cell transplant. And a stem cell transplant will fix the blood system, but it won't fix the pancreas or it won't fix the liver. And so the goal would be to try to develop these animal models, to develop means of either chemoprevention, whether it's taking a drug like metformin, and I'm just throwing that out, I'm not saying that there's any data for that, or to have more targeted therapy. And we're collaborating with the Italian group to look at their very promising but still very early data on trying to trick the ribosome into making a more functional Shwachman-Diamond protein through a method called nonsense suppression. And what we've done is just created the Zebrafish model, whereby we can actually test nonsense suppression in some of the drugs that the Italian group have developed. So, I mean, ultimately, our goal is to learn more about the biology of the disease, to understand risk factors, as well as to develop an animal model that can be useful to screen for drugs. Drugs that can either prevent disease systemically or even be more targeted for a particular set of organs. So this is where we're at. We just recruited another postdoc for the lab that does work on zebrafish. And hopefully I'm writing up our data now that it's been published into a grant from the NIH. So hopefully we'll get funded from the National Institutes of Health so we can really zero in on the ribosome defect, as well as to develop more and more animal models, the zebrafish models, to screen for drugs that will trick the ribosome into making protein.
A lot of discussion in the past year about the ribosome and its role in SDS, and it's kind of interesting to hear. It's always I don't know the right word for it, but it's very encouraging to see the types of different research that are happening for this disease. It's not just one or two, it's every organ. It's obviously SDS is very complex and it's multi-organed. And so thank you. I, for one, appreciate you as a family who deals with this quite often. So,
Right.
And I know I always find the zebrafish very interesting. It's one of my favorite pictures to show people when they really don't quite understand. I'm like, but look at these fish. We all see fish. Look at this fish, normal fish, this fish, very small fish.
Yeah.
So it's definitely encouraging and helpful. And on behalf of Nicole and I, Nicole is not on the screen currently, but I did want to thank you for coming to share this new research tonight, and it's very important. And there's a couple of questions we missed that are kind of scrolled back up, so I'll scroll through and I'll email those to you.
Okay, anyone can write to me at Corey-Lab, COREYLAB at Yahoo, or CoreyS2 at ccf.org. The manuscript.
There it is.
What? Oh, there we go.
Your email's going across the bottom of the screen.
Yeah. And I'd be happy to send the manuscript. It's written in dense scientific language. And if anybody has any questions, you can look at the pictures and then you can ask questions. If you read the sort of the first line in each paragraph, that's sort of the topic sentence, but then it gets into the density. And so I'd be happy to answer any questions. And in turn, I would be very much interested in hearing more about individuals' experience with liver disease, with any eye abnormalities, or any sort of organ issue. Then we can take a look and try to drill down in the fish. And, as I said, our goal ultimately is to try to identify who's at risk for developing leukemia, a genetic test so we can identify those kids before the horses left the barn, and protect their lives by a transplant, or alternatively to develop a pill that people can take. Not perfect, but it's better than not having any therapy to an individual. And the goal is to try to make people as normal without any deficits as possible, and certainly to try to prevent early onset of complications like liver disease.
Yeah.
And in turn,
very, very, very good goals.
Yeah. Okay.
Yeah. Thank you again for your time tonight, Dr. Corey. I really appreciate it, and being super patient through all of the technological issues that happened tonight that were not totally normal for us.
I don't know what happened, but anyway, everybody stay safe and sane and happy.
Thank you, Dr. Corey.
Yes, you too. Thank you, Dr. Corey.
Hopefully the hurricane
Bye.
Bye-bye.
Yes, I hope the hurricane stays far, far away.
Okay. Bye-bye.
Have a good night. Bye.
Good night.
Bye, everybody. Have a great night.